Okay, so what features of meiosis lead to genetic diversity among eukaryotes, and when do they occur? So we remember that there were two things that were normal events that happened during meiosis. So we're discounting mutations. Those are kind of abnormal events. Those are things that we haven't really programmed to happen. But the two things that lead to genetic diversity that are programmed into meiosis are crossing over. If you remember crossing over, that's going to happen during prophase 1 of meiosis. Zoom in more so you guys can see. That's probably better. Okay, so crossing over, that happens during prophase 1 of meiosis. Let's make it even bigger. And then the second process that happens is independent assortment. And that's where the homologous pairs will assign themselves to one or the other side of the cell when they line up down the middle during metaphase 1 of meiosis. Probably going too slow. That's just because I'm feeling slow today, actually. All right, what happens to tetrad during anaphase 1? So anaphase 1, so this is where so anaphase 1 meaning we're talking about meiosis, and anaphase meaning we're moving apart the homologous pairs from one another. And so the homologous pairs in proximity of each other are called a tetrad. So what we're doing to the tetrad is we're separating them. Separating the tetrad. Binary fission, the way bacteria replicate. Remember, they don't have any nuclei, and so mitosis, it alludes to the division of a nucleus. And so because bacteria have no nucleus, they actually don't undergo mitosis. And then cytokinesis, they just undergo binary fission. Which organisms reproduce through binary fission? Oh, actually, I shouldn't say bacteria. See, that was naughty of me. I would say prokaryotes. Because prokaryotes will encompass all things without a nucleus. That would be bacteria and archaea. So which organisms reproduce through binary fission? Bacteria, archaea. Remember, those are the two other domains of life. We've got eukarya, and then we have bacteria and archaea, which are prokaryotic cell types, which have prokaryotic cell types. What does haploid and diploid mean? Haploid means you have one of each chromosome number. One of each chromosome number. So your chromosome 1, you've got one from mom or one from dad, but just one. Or diploid, that means you have two of each chromosome number. How many total chromosomes are held in the nucleus of a human diploid cell? So in a human diploid cell, how many chromosomes, how many total chromosomes do we have? 46? 46? About 46? Would you agree that's 46? And so that would mean, because we are diploid organisms, we're going to have how many homologous pairs? 23 homologous pairs. All right. How many chromosomes are held in the nucleus of a human haploid cell? How many total chromosomes? We're going to have 23 total chromosomes. How many homologous pairs? 0 pairs. 0 pairs. Because a pair just implies that there's 2. So you're going to have one of every chromosome number, but 0 pairs. What are haploid cells resulting from meiosis called? Sex cells or gametes? What is aneuploidy and non-disjunction? Aneuploidy. What's aneuploidy? Can someone give me an example of aneuploidy?
It's when you have the wrong number of chromosomes, like trisomy 21 or something like that.
Yeah, so aneuploidy would be a different chromosome count for any chromosome number than one's parents. So example, trisomy 21. And so that's usually going to be the result of non-disjunction. So we know that, during anaphase 1, the homologous pairs should move apart from one another. They should separate from one another. But sometimes what happens is they do not disjoin, meaning or making it so that all of the chromosomes will go to one side. So we could say that, and this could be for anaphase 1. This could also be for anaphase 2. So we could say during anaphase 1 or 2, chromosomes do not separate. All right. Know the steps of cell cycle. What subphase is genetic material duplicated? Okay, so we'll just write it out. So we got G1 to S phase to G2 phase. S phase is where DNA is replicated in eukaryotes. All right. When chromosomes are uncondensed, what term is used to describe their form? When chromosomes are uncondensed, what term is used to describe their form? Chromatin. I think I might have something like that on the test. Chromatin. Rings a bell. What region of a chromosome holds the sister chromatids together? Centromere. What does the centrosome produce? Spindle fibers. What are those products used for? Oh, spindle fibers. Used for orchestrating mitosis or meiosis. So they're going to move shit around. They're the wires that kind of just move stuff around. What region of the centromere makes contact with spindle fibers? That's probably not going to be on the final, but it's the kinetochore. Be able to define mitosis and meiosis. Oh, this should have been like the first question. By now we should know it. Mitosis and meiosis. So mitosis is somatic cell replication. So this is going to be like a one-to-one. You're creating an identical copy. Or trying to, at least. And then meiosis, genesis of sex cells from germline cells. Not identical copy. Now we know that sex cells are going to be haploid. And then the fusion of two sex cells make a zygote. Well, actually, I think we're probably getting there at this point where we can fuse two of any sex cell. All right, be able to draw the subphases of my mitosis and cytokinesis. I'm not going to have you guys draw on the test. It's going to be purely multiple choice. Be able to use 6 words or less. Yeah, this is something that I normally, yeah, I'm not going to have you guys do that on the test. Define karyotype. It's going to be a snapshot of all, snapshot of all chromosomes frozen in metaphase. And this is usually what we'll do like when we're taking like an early genetic analysis of like an embryo. We'll define their, we'll get their karyotype to see, okay, do they have any missing chromosomes? Do they have too many of any chromosome? Know the difference between germline, somatic cells, and gametes. So germline are going to be the progenitors for gametes. Somatic cells are everything else pretty much besides your germline cells and your gametes. So, you know, our skin, you know, eyeball cells, all of your other cells that aren't related to creating gametes. And then gametes are sex cells. All right, chapter 9. What did Mendel spend most of his research trying to disprove? Probably won't have a question like that on the test, but what do you guys think?
That was Lyndon.
Huh? Oh, you're just listening. Or, yeah. Okay. Blended inheritance. Yeah, blending theory was one of them. To be even more accurate, though, it was irreversible blending theory. So yeah, you're on the right track. So irreversible blending theory, or inheritance, blending inheritance. Yeah, irreversible blending theory. Like I said, might not be on, most likely won't be on the test. Be able to differentiate between a gene and an allele. Gene and an allele. So a gene, that's going to be like, you've got genes for hair color, right? However, you don't just have one gene for hair color. Actually, let's use something else. Well, yeah, let's just use hair color. Yeah. So you don't have to just one gene, or you don't just have one allele for hair color. You actually have two. One that you inherited from your mom and one that you inherited from your dad. So the gene is kind of like the blueprint that gives rise to phenotype, whereas the allele is just one piece of the blueprint. I guess that, does that kind of make sense? So your allele, you know, like you've got in all of your cells 2 alleles, hopefully. Well, not hopefully, I shouldn't discriminate, but if you're a healthy human, you've got 2 alleles for every gene. You should have 2 alleles for every gene. So alleles are just the, you know, the pieces, and then the gene is the overarching gene. Okay. Know the difference between homozygous and heterozygous. So homozygous would be like RR or RR, so 2 of the same. Oh, it auto-corrected me. RR or RR, and then heterozygous would be RR. So hetero meaning different, or yeah, different, and then homo meaning same. Okay, know the difference between dominant and a recessive allele. So we could just use this example. Usually your recessive alleles are going to be masked by your dominant alleles, and so as long as you have a dominant allele present, the recessive allele will not be fully expressed. So, and then your, yeah, so dominant alleles be shown in phenotype regardless of if organism is homozygous or heterozygous. And then for a recessive allele to be expressed, will only be shown in phenotype if organism is homozygous for that trait, for that gene. So recessive allele will only be shown if it's homozygous for that gene. Know the difference between phenotype and genotype. So phenotype, physical characteristic. So just to use Mark as an example, it looks like, are you a redhead? Yeah.
When there was hair on my head, yes.
Yeah,
My
so you're a
beard.
redhead. So your phenotype is red hair. Your genotype, assuming that there's only one, we'll say that there's only one gene contributing, even though there's not, to having red hair, your genotype, because it's typically recessive, it would probably be homozygous recessive. Typically, I mean, genetics is kind of funky, but we're assuming that you're a pea plant.
My dad had black hair, and my mom has blonde hair. So
Yeah, see, that's where it gets funky. Yeah. Be able to infer a genotype based on a given phenotype. We kind of just did that. I mean, even though we were wrong, I mean, or we may have been wrong, but we could infer your genotype. We're assuming that you're homozygous recessive given your phenotype. Know the definitions of the examples. Okay. So polygenic, this is where I would usually start. So polygenic is where you have multiple, and this is where hair color kind of comes into play, even like skin color. So multiple genes contribute to a single phenotype. And then you can move on to pleiotropic, because we usually go from left to right. So poly, it starts with poly, which means many. So many genes contribute to one phenotype. And then the leftovers are pleiotropic, which is the inverse. So we've got, what is it? One gene, because we usually, when we're going from left to right, we also go from gene to phenotype. So it just moves along in line with that. So with pleiotropic, we have one gene contributes to more than one phenotype. Like with redheads, another example using redheads, typically if you have red hair, you'll also have freckles. Do you have freckles? You have freckles?
yes.
Yeah. So that's one perfect example of pleiotropic. One gene is going to contribute to more than one phenotype. Okay, incomplete dominance. So that's going to be where you actually get an intermediate phenotype. So if we breed a white flower and a red flower, we'll get a pink flower. So white, we'll just put here white plus red equals pink, because it's in between. Now codominance, the most common example that we'll use is blood typing, but just to piggyback off of this prior example, we have a white flower plus a red flower is going to be red plus white splotches. They're going to be, oops. Oh, red plus white splotches, yeah. So they're going to be equally expressed. And then epistasis, this is where one gene dictates the expression of another. So if you have the presence of one, or actually, let's just say, like you have your homozygous recessive for what is it? Albinism. What it does is that gene will mute the expression of all of the other pigment genes. All right, be able to do a monohybrid cross. Not gonna have you guys do this on the final because we're not, or at least I don't remember having any pictures of something like that. So, okay, cool. So we made it through, now we made it through 75% of the class. Oops. Is it this one? This one? Study guide 4. There we go. All right, be able to, oh, let's make this bigger so you guys can see. All right. So be able to match the contributions made by Aristotle, Buffon, Lamarck, Malthus, Wallace, Darwin. I'm probably just gonna focus on Darwin just because he was the main one who contributed. I like to go through the history during it, but when we're like recapping everything, going through the final, like trying to recall everything in the final, I want the takeaway to be at least that Darwin had contributed a mechanism for evolution. So you don't have to know the entire history. So Darwin provided a mechanism for evolution. Be able to, and he wrote the book, The Origin of Species. Be able to describe natural selection. Okay, let's try to do this in bullets. And this is probably asked multiple times too, just because, I don't know, kind of disorganized with this sometimes. So be able to describe natural selection. So variance, variance occurs in a population. Variance is genetic, which means it can be passed down. Some variance contributes to the survival and or reproductive success of the organism, we'll just say of organisms. Some variance contributes to survival and or reproductive success of organisms. So some genes may just allow you to reproduce a lot, and in some populations that's all that matters. If you can reproduce a lot, then you're successful. But mostly, if it contributes to survival, then, well, actually both of them are going to yield change. So some variance contributes to survival and or reproductive success of organisms. The individuals containing these heritable traits, these heritable traits that contribute to, we'll just say fitness, will produce more offspring, right? If you have like strong genes, you're going to produce more offspring. And so then the offspring, actually, then from here we can conclude, because the individuals containing these heritable traits that contribute to fitness, they reproduce more offspring, and we'll just say passing on those genes, that would lead to a shift in the population. So population will evolve over time. I kind of tie natural selection and evolution together, so it's probably going to ask for evolution as well. So yeah, that's what I would roll with here. Okay, be able to recognize the hierarchy of classification from most inclusive to least inclusive. Yeah, so let's just start with population. Did I say from most inclusive to least inclusive? Oh, haha, tricky. So most inclusive is everything, biosphere. A little bit less inclusive would be an ecosystem. A little bit less inclusive would be a community, which is going to contain various populations or various species, and then we've got a population. When we're talking about ecology, this is as far as I'd probably go. So population, that's going to be a population of organisms of all that all share a species. What's the ship that Darwin sailed on? What's the HMS Beagle? We don't need to know dates and stuff. I'm not going to test you guys on dates or his ship or whatever.
Yeah, the last one I had the "Do kids prefer candy over fresh green salad" written down. Is that?
Oh, yeah, I guess we could roll with that. Okay. Yeah. Well, okay, so we're talking about hierarchy of classification. Yeah, let's roll with that. Because that's when we're classifying. I guess yeah, there's multiple ways we've actually classified things in this class. Okay. Good call. So we got domain, which is don't. We'll start with the so we'll say don't, or what, there would be no apostrophe there, right? Don't, or yeah, there would be apostrophe. Don't would be domain. Don't kids, right? Kids. That's going to be kingdom. Prefer. It's going to be a phylum. Candy. It's going to be class. Over. It's going to be order. And fresh. It's going to be family. Green. Now we're getting into binomial nomenclature territory. Genus. And then salad. Species. Also, I just posted the final extra credit assignment for you guys to do. It's pretty simple. It's just you could use either Twitter or Instagram, and all I want you to do is post an organism. It could be any organism, a picture of any organism, and I want you to use binomial nomenclature somewhere in there. So you can just define it. You can take a picture of your dog, and what's the genus and species of that. Just make sure that they're italicized or underlined. And I know that these platforms sometimes don't let you do that, so you might have to copy paste from somewhere, like something that converts it to italics. And then just include a fun fact about that genus, species, that organism that people may not know. What is a fun fact about dogs, or the that specific type of dog, or that specific type of cat, or that specific bug that you found. All right. So know the general, and the thing is, you might have to get specific because there are specific species of cats. There's specific species of dogs. So they're not all going to be the same. Know the general tenets within the origin of species. Oh, okay. I was giving you guys the describing natural selection to you guys. It's derived from the tenets of the origin of species. That's the mechanism that we were provided with in that book. Define binomial nomenclature and be able to recognize genus and species from specific names. So genus is going to be listed first. Species is going to be listed second. It's going to be lowercase. They're both going to be italicized. So Homo sapiens. So our species is sapiens, and our genus is Homo. What are some pieces of evidence of evolution? I don't think I have you guys list the stuff, but we could just rattle some off. So fossil record. We could go to DNA, so biochemical ties, vestigial structures, embryonic similarities. Am I missing any? Fossil record, DNA, vestigial structures, embryonic similarities. Do I have any missing? Yeah, no. Okay, well, if I am, then I won't test on them. All right, so define natural selection. Okay, here we go again. Yeah, I knew this was gonna happen. Natural selection, you can kind of just group all this stuff into one big chunk. Because we got evolution through natural selection. Actually, hold on. Describe natural selection. Okay, I defined it. How do we describe it? I guess we would have to simplify it even further than this. Describing natural selection. Things, and I think the biggest bullet would be things that have heritable traits that contribute to their fitness produce more offspring, passing on those genes. I think this is the best summary of it. Things with heritable traits that contribute to fitness will reproduce more offspring and pass on those genes that contributed to their reproductive success. That's how you get change over time in a population. Define each of these three outcomes, each of the three outcomes of natural selection. Each of the three outcomes of natural selection. So we had stabilizing. Who can give me the other two? We have stabilizing, so that's when everything converges into one. We had two more. One of them starts with a D.
Directional?
Yeah, directional.
Directional.
And then what was the other one? Oh, they both start with a D. Disrupting. Disrupting. So stabilizing is where you'll get, we'll just say the medium phenotype prevails. And then directional is where we'll just say, you go from one extreme to another. And then disrupting is where both extremes prevail. Disrupting is where you get divergence. Usually this could possibly lead to speciation. This one probably has the highest chance of leading to speciation within a single population because you get divergence to both extremes. And that could just be because you have different environmental conditions just geographically. And so you could come from the same species, but some have different selective pressures than other, just depending on where they're located. It could even be, within, a few miles. There's different environments, different ecology, and, within a few miles, and you could get a disrupting process of natural selection occurring there. Directional is when you go, so the peppered moth is an example. That's where you move from one extreme to another. You go from, white with black peppered marks, and then you go to black with white peppered marks. So one extreme to the other. And then medium phenotype. So this could be, like you want a medium-sized tail because you want to use it for stability, yet you don't want it too long, or you don't want it too short because you use it for stability, and yet you don't want it too long because you don't want it to drag you around or create drag when you're running to catch prey. Okay, species. We'll just say organisms with, we'll just say similar DNA and can produce, so the main term here is can produce fertile offspring. Fertile offspring. So if you have close enough DNA and you can produce fertile offspring, which is just offspring that can also reproduce, then you could be considered a part of the same species. Taxonomy, phylogeny, systematic. So the one I'm going to probably test on the test is taxonomy. So taxonomy is the naming system. I got to fact check myself on that. I think that's what I had on the test, so we'll just roll with that. And if not, I'll just go back and change the test and make it taxonomy. All right, what's sexual selection? So that's going to be this is something that may actually kind of go against natural selection. So this isn't necessarily something that's going to contribute to survival. This is going to be something that just contributes to something's ability to reproduce, and that could just be like a peacock. You know, peacocks. What is what's their like main called? Their their display. Peacock's display. You know, they're going to get bigger and brighter over time because, you know, the female peacocks like big shiny things, yet that's not going to contribute to their survival. But if they can like get off a nut before they die, then they're going to pass on those genes. So that's sexual selection. Humans, that's where we're at. That's where we're at as humans. The more we reproduce, the better. Or the more selectively fit we are. All right, so know the difference between allopatric and sympatric speciation. Allopatric and sympatric speciation. So allopatric, that's going to be due, so this is due to geographic separation. So this is speciation. So speciation is the genesis or creation of a new species. So allopatric due to geographic separation. Am I even using, is it to separate? Do you spell that differently? No? Okay. So allopatric is going to be due to geographic separation, and sympatric speciation. That's where you actually, and this is going to happen really commonly in plants, and actually we like kind of engineer this stuff. And this is where you'll actually be within the same geographical region, and you'll be within, you know, the same ecosystem, but you actually get speciation, and usually in plants this happens from one generation to the next. So you have a mutation that produces a bunch of triploid stuff. Now those triploid things can mate with each other. But they can't mate with their parents. And so you've created a whole new species, and just like that. So that's going to be no geographic isolation or separation, yet a new species is formed. All right, what does fitness mean? So fitness is one's ability to reproduce. That's it. When we're talking about in terms of natural selection, so one's ability to reproduce. You have high fitness if you were able to create offspring. Know the three main domains of life. So that's going to be eukarya, bacteria, and archaea. General characteristics of each. Well, we know that eukaryotes, they're going to have nuclei. These two will not have nuclei. Bacteria will typically live in more temperate environments. So like more calm environments, the pH isn't too low, the pH isn't too high, the temperature isn't too low, the temperature isn't too high. And then archaea, those are going to live in extreme environments. And the way we can tell the difference between bacteria and archaea, besides the fact that archaea are living in extreme environments, is genetically. So we'd have to look at their genetic code, and then we could see the divergence just from looking at the genetic code. But for the most part, archaea will live in extreme environments. This could be even like high sulfur environments, or just like, you know, an abundance of things that would normally be toxic to bacteria. Okay. What is the peppered moth story? That was directional selection, remember? What type of selection? Yeah, this was directional selection. I'm not going to have you guys reiterate the whole story. We did like an extra credit on it, or a lab on it or something. I think we did a lab on it, and then it was on the test too. But what are some reproductive barriers? I'm not going to have you recall those. I mean, I would say that, you know, one of the biggest ones is geographic isolation, which leads to allopatric speciation. What level of organization does descent with modification act upon? Population. So Evolution does not apply to an individual, it applies to a population. All right, we're getting closer. And then we have one more after this. So what's the rain shadow effect? We've got, this is going to be ocean, the side of the mountain facing the ocean will receive more rainfall than the side facing away from the ocean. So we actually see this here. We'll see it in California too. We have, you know, the coastal region of California is like very temperate. It's got like perfect temperature. Lots of things can grow there. And then on the other side, like towards central California, it's just all desert. That's just an example of the rain shadow. And that's because the mountains are so high that the clouds containing all of the the rain clouds can't pass through the mountains. And so you'll have rainfall on one side and then the other side is left kind of dry. What causes seasons? That's going to be the tilt of Earth remains the same as it rotates around the sun. How much of Earth's water is fresh water? It's about 1%. That's just more of a fun fact. I probably won't test on that. Or maybe I will. I don't know. That's easy. You guys know that anyways. Know the difference between population size, population density, and dispersal patterns. So these are kind of like contained within the terms themselves. So population size would be the size of a population. So this could be like numbers, the amount of within a population, amount of individuals. Now density, this is going to be size of population or amount in a given space. By definition, that's what density is, amount of things per space. And then dispersal patterns, this is the only one that's, I mean, it's still kind of contained within the terminology, but dispersal patterns would be, you know, how things are dispersed. So you can have like clumped, random, and then some people like to use the term homogenized. So like an even distribution. Which biome is the highest contributor to oxygen on Earth? Marine. Be able to identify an accurate food chain. Okay, so usually when we're drawing a food chain, we go from the first order, or the producer, so the primary producer. And I'll actually write it out so we can hit multiple birds. So we have primary producer, so it's going to be our photosynthesizer. They're kind of what hold up the entire ecosystems of the world as our primary producers. What kind of primary producers live there? That's what allows you to, that's what yields the food for everything after that. So primary producer, after that we have primary consumer. So I'll just write herbivore, or, you know, omnivore, and omnivore. And then after that we'll have our secondary consumer. So in the simplest sense, that would be a food chain. So this could be like grass. If there's grass outside, your primary producer would be like bunny, and then your secondary consumer would be like coyote. So the arrows move in the direction of energy flow. So your primary producers are giving the energy to your bunny, and then your bunny is giving the energy to your coyote. What happens to chemicals in an ecosystem versus energy? So chemicals are recycled, and then energy is eventually lost to heat. So energy has like kind of a linear path, which eventually dissipates with every conversion. We know that the second law of thermodynamics says that every time you convert energy from one form to another, you lose some to an unusable form of heat. So energy will eventually be lost to heat. However, the chemicals, they don't dissipate into thin air. So the carbon, the carbon in the CO2, which becomes sugar, which gets integrated into your body, becomes part of like a fatty acid chain, and then you die, and that carbon just sticks around. Is recycled or cycled. Chemicals are cycled. Okay. Be able to identify instances of commensalism, mutualism, parasitism. So commensalism is going to be where one thing, and I'll just put a 0, one thing benefits, so it's positive, and then the other thing is neutral. Nothing happens. Remora fish are actually a good example of this. They're like these fish that will hang out and like latch onto the side of sharks. So sharks will go around and they'll kill shit, eat it, and they'll leave kind of scraps behind. And remora fish will then unlatch from the sharks and then, pick up the scraps and then attach back onto the sharks. So it doesn't really matter for the sharks because they're not, they're not consuming more, they're not taking away from the resources of the shark. However, they're benefiting. Mutualism, that's where both things will benefit. So lichens are a great example. Or we found that ants and aphids are another example. Lichens is a symbiotic relationship between algae and fungi. So fungi provide the housing and the structure for the algae, the protection for the algae, and then the algae provide the fungi with energy because they're producing sugars. And we got parasitism, and so this is usually grouped along with predation. So this is where one thing will benefit and the other thing will not. So you could think of it in the classic terms of a parasite. A parasite will live within a host, most of the time live within a host or be dependent on the host, and it benefits the parasite. However, the host is usually suffering some kind of consequence as a result. And then predation is just we could group it in because the fox eats the bunny. It does not benefit the bunny to be eaten, but it benefits the fox to eat the bunny. All right, which mineral contributes to eutrophication? What is eutrophication? So we could define eutrophication, So the mineral that contributes is phosphate, or phosphorus. Phosphorus contributes to that because that's usually along, within all of most of our fertilizers. The main component is nitrogen. We know that plants like nitrogen, but we also throw phosphorus into that. And we fertilize like a field of crops. It rains. That rain moves that water containing, the micronutrients within the fertilizer. It moves it to bodies of water. And so you move all that phosphorus to a body of water, for example. Algae are something that really thrive off of phosphorus. And so what happens is algae will bloom. We get an algal bloom. And so you'll get a bunch of algae covering the surface of water because they really like phosphorus. And as a result, we know that, within lakes and ponds, we still have plants living underwater. We still have fish that eat the plants. And we still have, there's a lot of biodiversity typically within bodies of water. Well, when you get that algal bloom over the surface, you're blocking light from penetrating the water as well as blocking oxygen from diffusing into water. And so what happens is the decline in biodiversity. So eutrophication would be, a lake essentially becoming more of like a wasteland or like a grave of many things underneath the algal bloom. Bacteria in the soil play a key role in recycling nitrogen. All right, so identify each of the terrestrial biomes. All right, well, I'm just going to do this. I'm not going to write this all out. So conifer forest, This is going to have a low biodiversity, but many living things, but low variation. So usually conifer forests, or actually to to define this even better, or to refine this, we would say taiga. T Taiga, which is kind of like a subcategory of conifer forest, is going to have very low biodiversity, but many living organisms. And we know that conifer, you could associate the con in conifer with cone, and all of those trees are shaped like cones. So you could think Christmas trees, they're shaped like cones. They have a pointy top, and they kind of, feather out towards the bottom. So those are the types of trees you're going to find in a conifer forest. And then broadleaf trees, those are going to be the trees that change colors with the seasons. So we see a lot of that here. And then temperate grasslands, that's where we're going to find sheep, a lot of grass, and not very many trees at all. Savanna is going to be usually closer to the equator. You're going to get extremely hot temperatures. You're going to get rainfall, and you're going to have a lot of grass, and you're also going to have some trees. But the most important thing, or the most distinguishing factor, is that they have the largest herbivores. I would say the largest terrestrial herbivores, the largest terrestrial animals, largest terrestrial, plants, or not plants, animals on Earth, terrestrial. And then chaparral, those are going to be usually coastal regions with salty soil. They're going to have seasonal fires, which aren't necessarily a bad thing, because a lot of the plants have adapted to fires, and so they're able to survive despite the fire, because they still have deeply embedded plant stuff in their roots deep in the soil, and so they can sprout up after the fires. But they'll have seasonal fires, and it's a great place for growing grapes. Desert, we know desert is going to have very little vegetation, but you're going to have very little rainfall. You're going to find camels and cacti. And then your tundra, that's where you're going to find your permafrost. So this is where you get, water. Water will soak into the soil with, grass or whatever, and it forms this permanent sheet of ice within that soil. And what that does is that prevents any plants from digging roots deeper into the soil. So we call that permafrost. And then polar ice caps, that's where those are pretty much the cold deserts. The very little vegetation, but instead of like sand dunes, you get like glaciers and lots of ice. You have polar bears and penguins. All right. So what happens to energy availability as trophic level increases? Trophic level increases, energy availability decreases. Carrying capacity. Population size and area can hold or can support. No, a producer, primary consumer, secondary consumer, tertiary consumer. We kind of went over that earlier. Your producer is going to be your herbivore, or not your herbivores, your photosynthesizers. Primary consumers are going to be the herbivores. Secondary consumers are going to consume the primary. Consumers and tertiary are going to consume the secondary.
Hey, Kyle.
I didn't go. Yeah.
You're at 55.
Yeah, I'm just going to keep going. Do you have what's it called today? The thing?
Yeah, we do, but I'd rather people stay here and do this than we'll
Okay.
just call this SI session.
Okay, cool. Yeah, I'm just gonna keep going until I finish. I just have that last snippet of stuff at the end.
Okay.
Yeah, and I'll post this as soon as I'm done. All right, so boom bust cycle. That's where you get like the oh, we saw it. I think we used foxes. I think we used foxes and snow hares, or no, did we use lynxes and hares as an example? So we know that lynxes like to eat hares. And we know that lynxes are kind of dependent on the hares for their food. However, we could start, like at ground zero, and you have a large population of hares, let's say you have lynxes that haven't really met. They're migrating around, and then they find this large population of hares that live in a specific region. And so to start off, your hare population's astronomical. You've got a ton of hares because they have no selective pressures. They don't have predators trying to eat them. However, the lynxes migrate over and they start consuming the hares. And so what happens is the hares population will hit a sharp decline, and those lynxes, well, they're fed. However, now there's less hares, and your lynxes have nothing to eat. So you're going to have a decrease in your lynx population. With that decrease in lynx population, you'll have a boom of hares reproducing because there's less lynxes to consume them. And as a result, your lynxes will consume that large population of hares, and their population will rise. And then you just have this staggered boom and bust, one after the other, within the two different populations, or you could say within the community. Which type of growth indicates limited resources? That would be logistic growth. Right? Wait. Yeah, logistic growth. Compare interspecific competition, intraspecific competition, and give an example of each. So inter, think internet, think species interacting with many other species, so that'd be interspecific. Intra would be competition within one's own species. So interspecific is competition between other species, so the hare and the lynx have interspecific competition. And let's say, the lynxes are competing to consume the hares, and they're competing amongst themselves to consume the hares. That would be intraspecific competition. Keystone species, what's its role in an ecosystem? So keystone species is a species that will hold a population together. Species that will hold a population together. Role in an ecosystem, or so it by hold a ecosystem. So species that will hold an ecosystem together. And they're doing that by consuming things that could potentially become overgrown. So the example that we used in class was starfish. We know that starfish are a predator for mussels, but if we remove all those starfish, what happens is the mussels will become overgrown and it'll drive out other species, so it'll lower the biodiversity. So that would make starfish, the predator, actually a keystone species. Remediation is picking up trash, essentially, and restoration is bringing back an ecosystem into its historical existence, what it looked like, what it felt like, what it was at some point in the past. Okay, let me find the last one. All right, we'll roll through this really quick. X-ray crystallography, transcription, translation, start codon, stop codon. Wow, made this hard on myself. So X-ray crystallography, this is going to be, without getting into too much of this is going to be visualizing the way we visualize small things. We'll just say that. This was what Rosalind Franklin and yeah, this is what Rosalind Franklin used this to visualize DNA. Okay, transcription. So transcription, that's going to be, it's going to happen in the nucleus. So happens in nucleus, and this is going to be converting our DNA into mRNA. That's going to be, we know that that's orchestrated by RNA polymerase. Translation, that's going to occur in the cytoplasm. That's going to be converting mRNA into protein. Start codon. The reason why I have this in here is because of the term codon. We know that when we're talking about codons, we're talking about translation. So this is going to be where the, we'll just say where the ribosome begins translation of a protein. Now we have promoter, so there's no codon there, but that's where this is going to be what recruits. This is going to be a sequence on DNA that recruits RNA polymerase. And we know that RNA polymerase is what initiates and pretty much conducts transcription. Oh, and a stop codon. So a stop codon, sorry. I skipped over that. Stop codon tells ribosome to stop translating. Okay. RNA polymerase, enzyme that conducts transcription. Peptide bond, that's what we're going to find between each amino acid. Between each amino acid in a protein. This is what binds your proteins together. This is what you're using. This is what you're, every time you bring in a new amino acid, you're going to bind it to another amino acid using a peptide bond, or the peptide bond is what you're forming between them. Ribosome, this is going to be the thing that translates mRNA into protein, and it's going to be usually composed of protein and rRNA, ribosomal RNA. And so the next one is rRNA, ribosomal RNA. That's what it is. tRNA, that's going to be transfer RNA. And this is going to be what brings amino acids to the ribosome. Okay, what did Chargaff, Watson and Crick, and Franklin and Wilkins contribute to the discovery of DNA structure? I don't know if I go that far. Huh. We'll talk about it anyway. So Chargaff was the ratio guy. So to further define this, A should equal T, right? A should equal T, G should equal C, if you were to take a DNA sample. Watson and Crick, they kind of deduced everything, used available information to deduce structure of DNA. Franklin and Wilkins, so that's going to be Rosalind, Franklin, and Wilkins. They use X-ray crystallography. So we'll just say visualize DNA using X-ray crystallography. Cool. Know which bases are purines and which ones are pyrimidines. Ooh, that's a pretty good one. I don't know if I'm gonna test on that one, but do you guys know? There's got to be a way to remember this. I don't know. I don't remember using any. Let's try to come up with one right now. Let's try to come up with like an acronym or something. I think purine, like pure as gold. I think that was one that I might have used. Well, anyways, I'm not going to test you guys on it, but I'll just, it might help you guys in the future. So purines, they're going to be adenine and guanine. Pyrimidines, it's going to be C, T. I think uracil would actually fit into the, yeah, uracil fits into that category. So purine, so pure, and G is gold, so pure as gold. Maybe the as, the A in as, pure as gold. And actually, if you could just do that, pure as gold, purines pure as gold, then you could kind of deduce, okay, so the remaining things are going to be pyrimidines. Maybe that's what I used. I don't really remember. I mean, pyramids, you could, if you tilted the C to its side, it would kind of make a, no, it's not going to make a pyramid. All right, yeah, pure as gold. We'll just roll with that. Know the 4 features that describe the structure of DNA. Is it 4 features? Let's just see. We got the 5-carbon sugar, phosphate, 5-carbon sugar, phosphate, plus phosphate backbone. We've got the nitrogenous base. We know that DNA is going to be anti-parallel. It's going to be a right-handed helix. It's probably more than that, actually. Right-handed helix. Right-handed double helix. If I think of another one, I'll come back to it. Be able to reconstruct the events of protein synthesis from nucleic acid to functional protein. I'm not going to have you guys do that on the test. Let's go with this, though. Transcribe a strand of DNA. Okay. So one thing that I could probably add here is if you see a 5' and then a 3', so like we'll just put it like that. Know that your answer, because this is going to be multiple choice, your answer should be the opposite. Know how to use the genetic code table. Okay, let me find it. Well, I'm going to give you guys the answer here. So be able to use, I'll just run through it one more time. This is actually, oh, that's not the one. Hold on. I'm going to try to find the one that I actually give you guys on the... one that I use. Oh, hold on. One that I use, oh my god, that sucks. Oh, well, I use this one on the exam. So we don't need this junk out here. But, so your first letter is going to be on the left side. second letter is going to be on the top, and then your third letter you're going to find within here. So let's just say, you know, we started with AGG. So we have A, then we go find this G, so G, G, ARG. So arginine. So you'd start off with arginine in the amino acid sequence. And then let's say the second codon, which is just a group of 3, was like CAA. So you'd have CAA glutamine. So you'd have arginine, glutamine. And I probably won't have you guys do any more than 3. I'm not gonna, because it's just busy work after that. I'm not gonna have you do busy work on the test. Okay, I just want to make sure that you guys know how to do it. What's the function of a spliceosome? Splices exons together, takes introns out. Know the makeup of DNA backbone? What holds the 2 strands of DNA together? So we already know that the backbone is going to be, the 5-carbon sugar and phosphate. What holds the 2 strands of DNA together? So when you're holding 2 strands of DNA together, you're going to hold them together via hydrogen bonds. And know which bases pair with one another. Know which bases pair with one another. So A to T or U, and C binds to G. Know how RNA differs from DNA. Okay, so I think the main thing I want you guys to know is DNA is going to have A, C, and G, and then RNA is going to have A, U, C, and G. I'm not going to have you guys get into the nitty-gritty of the difference in the molecular structure of them. Just know functionally how they're different. Know the different types of point mutations that can occur, and then know the difference between silent, missense, and nonsense mutations. I think I just asked the same question twice. So, silent mutation, no change in the end result, and know that silent mutation is going to yield no change in the end result. So you're let's say, and we could actually use the genetic code as an example. Let me find a different one. Hold on. Open up a new tab. No, hold on. Almost done, guys. Image a new tab. Here we go. So let's say, one of the amino acids was originally CCA. So CCA, proline. And what we did is we had a point mutation that changed that second C to an A. So it would make it CAA. So CAA. So what you've done is you've changed, oh wait, no, we're talking about silent. Sorry. Silent mutation. So you have CCA. So let's say we change that third amino acid, or third nucleotide into a U. So CCU. Both the same amino acid. They both code for the same amino acid, so no harm was done. There was no change. So that would be a silent mutation. A missense mutation would be if we actually got a change. So let's say it was CCA originally, and we changed that second amino acid to an CAA to make it CAA. So you'd go CAA. You change it from a proline to a glycine. That would be a missense mutation. Oops, sorry. Change amino acid. Now, a nonsense mutation, I like to associate that non in nonsense with no, or with stop, with no, you're no longer going to create your protein. So let's say it was, originally, I don't know, UAU, So we have UAU, so let's find UAU. UAU, tyrosine. So let's say we change that third amino acid to an A. So instead of UAU, it's UAA. So we got UAA means stop. So instead of actually creating a new protein by just splicing in a new amino acid, what we've done is we've created an early stop codon. So you stop producing that amino acid or that peptide. So premature stop codon is the key terms you're going to look for. Premature stop codon would be nonsense. Okay, so that's that.